Okay, welcome back everyone. So this lecture will cover most of the topics that are in your urinary system chapter in the textbook. I still recommend reading that chapter because there are some additional pieces of information that I'm not including in this lecture, just because if I did, this lecture would be an hour and a half long. So I'll try to get through this in as quick as a time as possible, but don't be afraid to pause if you need to take notes during any point. So um, what's going to happen is I'll talk about a small introduction, and then we'll talk about kidney functions, including glomerular, glomerular filtration, tubular reabsorption, and tubular secretion. So, the urinary system, or also called the renal system, is basically starts with your kidneys. And we have what is called the functional or the working unit of the kidney. There's billions of them, and they're called nephrons. And they're where the urine is produced. Then the urine will travel through the ureters into the bladder and then be excreted or basically exit the body through the urethra. Now, the urethra is slightly different in males and females. It's the only part of this system that is different between male and females. So give that a look in the chapter. I'm not going to go into depth about the differences in this video. So let's talk about kidney functions. The kidneys are the primary organs of the urinary system. They get a huge blood supply. About 20 to 25% of your cardiac output travels through from the heart to the renal artery. The functions of your kidneys are to maintain blood volume, maintain fluid and electrolyte composition through levels of water and levels of ions in the blood, um, maintain acid-base balance by controlling levels of the levels of protons, are basically H plus molecules, ions. They eliminate metabolic wastes and toxic substances. They do vitamin D activation, and they produce hormones, erythropoietin or EPO, which is the hormone that we learned controls red blood cell production, and also renin, which is part of that RAS system, RAAS, and that just deals with water and salt conservation. So, what is urine? I mean, I'm pretty sure we're all familiar with it, but what is it scientifically? It's a fluid waste product that's produced by your kidneys, and it's basically a filtration of the blood. Whatever needs to be returned to the blood gets returned to the blood, and whatever needs to be eliminated from the body stays in the urine. What it normally contains is water, some ions, of course, nitrogenous waste, and small soluble compounds. Composition will change as a function of blood and kidney regulation. So if things are going on in the body, composition of the urine can change. What it should not contain are blood cells, large proteins, and presence of either of those would indicate damage to your kidneys. So, nephron, it's the working unit of the kidney. It produces urine. I said there's over 1 million nephrons in each kidney, and it removes, exchanges, and adds materials to and from the blood to regulate blood composition. So first we'll ask ourselves, what stays in the blood? Well, you get blood that comes into the nephron, and then regulated blood comes out and returns to the body. And then what needs to be removed from the blood to stay in the urine? Well, you get blood in, it will remove the waste, and the urine will leave the body. So let's talk about what things stay in, what things leave. Okay, so here is what a nephron looks like in the body. And so it's a little bit convoluted right now, but I'll walk you through it, and hopefully it'll all make sense. So let's walk through the anatomy of the nephron together. Okay, so entering the nephron is a large blood vessel, and this large blood vessel is called the afferent arteriole. And the afferent arteriole branches into a ball of capillaries called the glomerulus. Okay, so here's the glomerulus. Then the blood that exits the glomerulus, we'll eventually go into here, but we'll talk about first, blood that exits the glomerulus leaves out the efferent arteriole. So here's the efferent arteriole, not labeled here for some reason. The efferent, so that's an E. So afferent arteriole brings it into the glomerulus, and starting with an E, efferent arteriole leaves the glomerulus. That blood continues to branch around the kidneys into a group of capillaries called the paratubular capillaries. These are all these capillaries right here that are branching around. These are called the paratubular capillaries. Now, constant exchange is happening through the paratubular capillaries. That's why they're so close to the rest of the nephron. So surrounding the glomerulus, let's go back here, surrounding the glomerulus is this large capsule, which is the start of the nephron. That capsule is called Bowman's capsule. So this is Bowman's capsule right here. And it's lined with epithelial cells and has a hollow center, okay? Here's Bowman's capsule with the hollow center. Together, the Bowman's capsule and the glomerulus is called the renal corpuscle. So R-E-N-A-L, renal corpuscle, C-O-R-P-U-S-C-L-E, renal corpuscle. So the blood enters the afferent arteriole, and it will first be filtered, and the filtrate will enter Bowman's capsule. From there, we get a series of these winding tubules that we first call the proximal convoluted tubule. So proximal because it's close to the renal corpuscle, and it is convoluted just because it's all twisty and windy. So we get the proximal convoluted tubule. Then as the urine or the filtrate basically travels down there, it goes down this long looping tube, and it is called the loop of Henle. The loop of Henle. So it's loop of H-E-N-L-E, the loop of Henle, and there is a thin part, which is the loop, part of the loop that goes down, and a thicker part, which is the part of the loop that goes up. Okay, and so the urine filtrate will travel through this PCT, or the proximal convoluted tubule, down the loop of Henle, and then back up the loop of Henle. From there, the filtrate enters to the DCT, or the distal convoluted tubule. So it just means it's a little farther away from the beginning of this this whole process. So proximal, PCT, loop of Henle, now into the DCT. And then the last stop is the collecting duct. Basically collects the urine, and that urine will then flow into the renal pelvis down into the bladder. And the urine eventually is just going to exit out the body. Remember that along this pathway, when we have this filtrate going along this pathway, there's going to be constant exchange back and forth between those paratubular capillaries. The paratubular capillaries will be exchanging back and forth. Either they need to get some stuff back that was pushed into the filtrate, or they need to add more stuff into the filtrate. And so that's what we're going to talk about now. So there's three main processes that are happening in the nephron. The first is glomerular filtration. Think big from small. It's the beginning, right when it enters the corpuscle. It's separation of cells and large proteins from the plasma, and the plasma will enter the nephron. Then we get tubular reabsorption, where valuable substances that are in that plasma are selectively returned to the blood from the nephron, and tubular secretion, where unneeded substances that didn't get in for the first time are then removed from the blood selectively and added to the nephron. And we'll talk about all of them in a little bit more depth right now. Okay, so glomerular filtration is a non-specific separation of large substances like cells and proteins from small substances like water, ions, and small molecules. Think like a pasta strainer. You're boiling some pasta, you dump the mixture of the water and the noodles into the pasta strainer. Excuse me. What's left is your pasta, the large molecules, and down in the sink or the pot, wherever you're straining it from, is the small molecules like the water and the ions. So think about it that way. Blood, of course, enters at the afferent arteriole and passes through the glomerular membrane, and that's where it's originally filtrated. Large proteins and cells stay in the blood and exit at the efferent arteriole, and the filtrate, which is the water, ions, the small molecules, they'll enter filtrate into the nephron tubules at the Bowman's capsule, into the PCT, down the loop of Henle, up the loop of Henle, into the DCT, and into the rest of the nephron. So we'll talk about something called the glomerular filtration rate, or GFR. The volume of filtrate formed by both kidneys per minute is termed the glomerular filtration rate, or GFR. The heart pumps about 5 liters of blood per minute under resting conditions. Think about that. That's approximately 20%, or 1 liter of that will enter your kidneys to be filtered. On average, that liter results in the production of about 125 milliliters per minute, a range of 90 to 140, and 105 milliliters per minute in women, a range of 80 to 125. That basically equates to a volume of about 180 liters in men and 150 liters per day in women. But 99% of that filtrate is going to be returned to the circulation by reabsorption, so that really only what you're making is about 1 to 2 liters of urine produced per day. But that's still a crazy amount. Think about how much a liter is. So GFR, this is going to be, we're talking about some sort of high-level stuff here, so just sort of follow along with me. GFR is going to be influenced by the hydrostatic pressure and the colloid osmotic pressure on either side of the capillary membrane of the glomerulus. And so if you remember when we talked about capillary exchange, it's sort of the same idea, in which hydrostatic and osmotic pressures basically force which way things will be filtered. Filtration occurs because pressure forces fluid and solutes through the semi-permeable barrier where the solute movement is limited by particle size. Okay? So, just like we said, this is where the big particles get filtered out. If you have a fluid that's happening on both sides of a barrier, both fluids would exert a pressure in opposing directions. Net fluid movement will be in the direction of the lower pressure, okay? So whatever area has lower pressure, we're going to get all of the fluid rushing that way. Remember that osmosis is a movement of water across a membrane due to a gradient in the amount of solute on either side. That creates a pressure called osmotic pressure, and that exists until the concentration of the solute is the same on both sides of the membrane. As long as that concentration is different, water will continuously move. So glomerular filtration will occur when glomerular hydrostatic pressure, right here, exceeds the luminal, or the inside, hydrostatic pressure of Bowman's capsule. There's also an opposing force, the osmotic pressure, right here, this 30 millimeters per mercury. That opposing force is the osmotic pressure, which is typically higher in the glomerular activity. Why is that? Well, in order to understand that, you have to look more closely at the microenvironment on either side of the membrane. You find that osmotic pressure exerted by the solutes inside the lumen of the capillary, as well as inside of the Bowman's capsule. And since the filtration membrane limits the size of particles crossing the membrane, the osmotic pressure inside the glomerular capillary is going to be higher than the osmotic pressure in Bowman's capsule. So remember, cells and large proteins can't pass through, and so that's the reason why. This means that red and white blood cells, platelets, albumins, and other proteins that are too large to pass through the filter remain in the capillary, creating an average colloid osmotic pressure of 30 millimeters per mercury within the capillary. Okay, so that goes this way. The absence of proteins in Bowman's space, right here, results in an osmotic pressure near zero. Thus, the only pressure that's moving fluid across the capillary wall into the lumen of Bowman's space is hydrostatic pressure. And that's sufficient enough to push the water through the membrane despite the osmotic pressure working against it. So the sum of all the influences, both osmotic and hydrostatic, results in a net filtration process of just 10 millimeters per mercury. So basically what that means is this is a very small net outward pressure, but it's enough that all this filtration will continue to happen. Now, what you should take away from this is that this is a really small amount of net pressure. So basically, you should realize that minor changes in the osmolarity of the blood or changes in capillary blood pressure could result in major changes in the amount of filtrate that's formed at any given point in time. Luckily, your kidney is capable and able to cope with a wide range of blood pressures. In large part, that's because you have smooth muscle. And when you stretch smooth muscle, it contracts. So when your blood pressure goes up, smooth muscle in the afferent capillaries will contract, so you'll limit an increase in blood flow and filtration rate. You don't want to be filtering way too much because it's not going to be able to handle it. And conversely, when blood pressure drops, the same capillaries will be able to relax and maintain blood flow and filtration rate. So the net result is a relatively steady flow of blood into the glomerulus and a relatively steady filtration rate in spite of what could be significant systemic blood pressure changes. And I know that's a lot of information, guys, but basically all you have to remember and all you need to really take away from this is that filtration is always going to continue as long as the net pressure is greater coming from the outside into the nephrons. And as long as you take away from that, from this course, I'm going to be happy. Okay. So that was filtration. Now let's talk about tubular reabsorption. And that's the selective process of returning needed substance from the filtrate back to the blood. You start with the proximal convoluted tubule. Materials are going to be selectively transported back to the peritubular capillaries, which is basically the blood, and to be returned to the body. But excess materials and waste are going to stay in the filtrate and, of course, exit as urine. What kind of things go back to the blood? Well, how about 99% of the water, 100% of sugar, and 99.5% of salt? Excess ions, though, urea and toxins, all that stuff is going to remain. So it's really, really important that we reabsorb this stuff, because if we just got rid of all that water, we'd go dehydrated extremely fast, very, very fast. If we got rid of all that sugar, we would be, our cells would be starving. And if we got rid of all that salt, basically what you'll see is that our electrical activities would be thrown off. Where do we need electrical activities? Well, anytime you've got neurons firing, those are electrical activities going on, okay? And then lastly, tubular secretion is the process of actively removing substances from the blood and putting them in the filtrate to be eliminated. It's an additional step to ensure that any waste and excess is eliminated from the blood and the blood is balanced in every way. Things like proton ions or hydrogen ions, that helps with, of course, if you remember, like pH, so acid-base balance. Then, of course, K plus ions, that helps with our sodium and potassium pumps. K is, of course, potassium. And then, of course, organic ions. So, like I just said, proton secretion or hydrogen cation, remember cations are positive ions, it's regulated in order to maintain acid-base balance, and that actually normally causes your urine to be acidic. Or if your blood is around 7 or neutral, your urine is actually more acidic where it's towards 6 pH. That occurs in the PCT, the DCT, and the collecting duct, and it's balanced with bicarbonate reabsorption. If you remember, bicarbonate reabsorption will make your blood more basic. Okay, so that's just that's something to keep in mind. Then we talk about potassium secretion, and that's regulated to maintain electrical gradients, especially in the heart, where we have to be firing electrical signals at all times, and to maintain sodium-potassium pump activity in the kidneys. If you remember, those are ways in which we can do active transport of ions into and out of cells. This occurs in the DCT and the collecting duct, and secretion of this is stimulated by aldosterone. And then lastly, tubular secretion of organic ions includes hormones or foreign compounds like pesticides, pollutants, food additives, and drugs. There's two types of non-selective carriers that carry these ions, either anions or cations. anions being negative ions, cations being positive ions. The consequence of them being non-selective, however, is that if you had two drugs in your system that need to compete for the same carriers, one drug might not be fully eliminated. And that can cause problems because you obviously need to get these drugs eliminated from your system at some point, and it also could cause drug interactions. And so that's one of the reasons, before I get into that, that's one of the reasons why doctors will want to be able to test your filtration rate, because they want to know if you are going to be healthy enough to excrete these drugs that they could give you that if they stayed in your system too long could become toxic. So we've talked about how urine gets made. Well, what's the big shebang? What's the big show? And that is, once urine is produced in the bladder, it's stored in the bladder. But how do we empty the bladder? And that's called micturition, or bladder emptying. And basically, what happens is your bladder just stores urine until it starts to stretch, and the receptors in the bladder wall sense about 250 to 400 milliliters and signal to the parasympathetic nervous system via your spinal cord that it's time to go, right? It's literally time to go. The parasympathetic nervous system stimulates bladder contraction. It'll open up the internal urethral sphincter. You'll feel the urge to urinate, and then you voluntarily control the external urethral sphincter, which is skeletal muscle. And this is just something I'm sure we're all kind of familiar with, but the yellow pigment in urine is called urochrome, and urochrome is one way in which you can determine the health status based on your urine. So, of course, the more dark and more yellow, the more dehydrated somebody is. And also things like if it has a red tint to it, that means there's some blood in the urine. That could point to a number of problems. Also, if you have like a dark, almost like cola, like a cola color, that can point to types of cancers and things like that. So the color of your urine can point to a number of health factors as well. And so with that, that's going to be the end of my lecture for this chapter. Make sure you check out all the other videos that I post for this chapter, and I'll see you in the next one.